Hi everyone, here's some mnemonics I made for cell bio, which I hate. Okay, mismatch repair. Missile launched up. So MIS here stands for mismatch repair. There's your image. Great. So mute S and mute L are going to find a nick, which is on a DNA strand, and then endonuclease is going to cleave it, and then DNA polymerase is going to fill in the gap. So, and launched because it's defective in Lynch syndrome. Base excision repair. This only makes sense with the picture. It's Zoolander. So berry gel application DAMSER. So like hair gel, right? BER, base excision repair. So DAMSER stands for deamination and cancer. So deamination being where your cytosine in your DNA gets changed into uracil because you lose an amine group. So that's not good. So the way you repair it is glycosylase comes in and removes it, and then you got an empty spot there. Endonuclease cleaves the 5' end, like lyase cleaves the 3' end, and then DNA polymerase fills in the gap, and then ligase seals the breaks. So that's just the sequence of bases. Or not bases, proteins that's involved. Okay, and this is a bunch of random junk. I guess most of cell bio is random and biochem, but this is a nice mnemonic I made back in the MCAT days. So it's for base pairs. Most people have other ways of memorizing this, or they just know it, whatever. But you spell guac counterclockwise, and you draw a chip with 3 lines at the top and 2 at the bottom, because your chip is wider at the base, because you're going to dip it into guacamole, right? So the horizontal tells you what is binding with what, and then if you want to know which ones are purines versus pyrimidines, I draw my Y's like this, right? So the largest line is this diagonal, so this is your purines, or pyrimidines, sorry. And I draw my U's like this, so the overall direction is like that, so these are your purines. Here is tRNA, and for the tRNA parts, I write data twice, and then I write try AA, like a subliminal message to myself, I guess, I don't know. So DARM binds aminoacyl tRNA synthetase. The tARM is where your ribosome binds, and then your AA, the acceptor stem is for amino acids, and acceptor is 3' ACZ over there, so that's kind of nice because that's the first 3 letters of acceptors. So this last one's kind of junk, I admit. Eukaryotic RNA polymerases, 1, 2, 3 RMT, and that's what it means. Oh, one more. Okay. Before I go into my larger one. So DNA polymerase makes 5' to 3'. I just imagine a mirror here because the whole DNA polymerase process is like mirrored, you know, how everything works, everything's split in half and then done again. So M looks like a sideways 3, S looks like a 5. So if you do this and then it goes across a mirror, it's going to come back in the reverse order. So that's how I remember that. So DNA replication, I made a visual mnemonic for this because I suck at it and I forget it, 3 seconds after I study it. So I'm going to walk through this, and you can use it if it helps. Here's topoisomerase, which is the ice top mountains, and they're way north of everything else here, relieving supercoiling. So then this is a whole military operation going on. You have a helicopter flying it, and this is HQ watching in the background. But you have a helicopter flying in, and it's going to split into leading and lagging strands. So your leading strand, he's going to be leading a prisoner out. You're just going to have one RNA primase, right? These are primate soldiers, I guess. So he's rescuing a single-stranded bound prisoner, right? Single-stranded binding proteins are going to be actually on both of these, but they prevent everything from rebinding when you're splitting it open. So on your lagging strand, you have multiple RNA primases, and then you have these double palm trees here, which are DNA polymerase 3s, which are going to be the eukaryotic version. Prokaryotes also have double palm gun, right? DNA polymerase 1 here. So DNA polymerase is going to bind to your primase, and they form an Okazaki fragment, which is the bazooka that he's pulling out. Okazaki bazooka. Some important fact about your DNA polymerase 3 is you can see the little treasure line going from it to a beehive to an X, marking the spot. So the X stands for exonuclease. DNA polymerase 3 has a 3' to 5' exonuclease activity, whereas DNA polymerase 1, he's got an X marked on his bullet. So it goes from 5' to 3', right? That whole direction for its exonuclease activity. DNA ligase, the liger here in the village, it's trying to defend it, is going to be joining Okazaki fragments. And then your origin of replication is your origami village way down here. So I used to get confused about where that was. That's like in the middle of the whole split. And lastly, telomerase, telephone, is in eukaryotes only. So there's a giant nuclear launch button here because they have a nucleus. And what it's going to do is add TTA-GGG, which is your dog tags, to the 3' end. So there's 3 dog tags. So this guy is learning that 3 soldiers have died. So that's why he's got the dog tags. So 3' end, it adds TTA-GGG so that you can live longer without having DNA mutations. You can have more replication cycles. And I rewrote all that. So that's everything. There you go.